Our final approach to biological evaluation of these complexes was the molecular docking studies. Molecular docking was explored to show the binding potential possibility of the synthesized compounds tested against several breast cancer related proteins. It is used to find the best conformation of the ligand binds to the active site of the selected proteins, and the binding pocket selected in the docking is 8 Armstrong. The binding affinities can be found in this table on this slide. Since we have chosen breast cancer cells to evaluate our molecules, we decided to perform molecular docking studies using protein structures that are responsible for this disease. So more specifically, we have used cyclin-dependent kinases and 6 inhibitors in association with endocrine therapy represent the gold standard for the treatment of estrogen receptor positive human epidermal growth factor, and receptor negatives metastatic breast cancer. Moreover, HER-VO overexpression is associated with more aggressive disease, higher recurrence rate, and shortened survival. So, since our metal complexes provided anti-proliferative activity against MTA and B231 cells, we wanted to investigate the specific mechanism. So we checked the binding affinities on LMTK3 protein, which the first vanadium complex had the highest binding affinity, while the second vanadium complex had the best binding affinities compared to other 3 molecules on CDK6 and HER2 protein targets. I will not bother you with details about what amino acid residues interact with these complexes. You can find it on these tables, as well as if the interactions were hydrogen bonded or van der Waals interactions, and etc. Okay, moving forward to see this slide of another work. Describe the use of vanadium 3, 4, and 5 complexes as potential candidates in drug screening with computer-aided chemistry for COVID-19. In particular, we use the example of Vita-TOC-DEA, which was transition state 5, and Vita-TOC-DPA, which was transition state 4. This paper has been published. And the amphiphilic vanadium complexes, we saw that they exhibited enhanced hydrolytic stability. All the compounds found to be cytotoxic for cancer cells in a previous study, so we checked them with in silico studies for COVID-19 as well. Actually, we perform molecular dynamics, density functional theory studies, and molecular docking studies with these molecules against the bovine serum albumin, human serum albumin, transport protein, 6M03, which was the COVID-19 main protease, and 6M71, which is the SARS-CoV-2 RNA-dependent polymerase, in order to see the binding affinity of these molecules to the COVID-19 proteins. As we can see from table 1, the calculated total energies of Vita-TOC-DEA, Vita-TOC-DPA, and VXN, a complex of vanadium 5 with the prenylated flavonoid, prenylated halcon xanthohumol, are in order from lowest to highest. So the value of the energy difference between here HOMO and LUMO orbital plays an important role in the stability and reactivity of the molecules. That's why we did this density functional theory studies here. Let's make it quick with the results. As we can see, VITA-DOC-D and VITA-DOC-DPA have similar reactivities and kinetic stability. The same is expected with vanadium xanthocumol complex, which is complex molecule designed based on the previous two complexes. Based on these results, we have used the geometry of the model tocopherol to design the vanadium xanthocumol molecule. Both are vanadium complexes with natural derived ligands, and the results confirm that. Finally, molecular docking method is one of the frequent referred structure-based design methods. We decided that because the ligand molecules will be screened as a drug candidate for COVID-19 to perform studies with several related proteins. I said before the 4 proteins that we used. Moving forward to the results, we saw that these molecules had high binding affinities, as you can see on this slide, with the COVID-19 proteins. Some of them with hydrogen bond contribution, some of them with van der Waals energy contribution. So specifically, the SARS-CoV-2 RNA-dependent polymerase, starting with the ligand molecule with the highest affinity to the protein, VBCAG is the first, followed by VITA-DOC-D, VITA-DOC-DPA, and vanadium xanthocumol complex. All the detailed information about the energy amounts of the ligand molecules through the different proteins, hydrophobic and hydrophilic interactions, can be found in this table as well. So before thank you, I show you here actually 2 main works. The one was the synthesis and characterization and biological evaluation of 4 complexes, 2 of zinc and 2 of vanadium, with amino substitute derivatives as ligands. And the last one, the second one, was an example of a variety of vanadium 3, 4, and 5 complexes from previous works of our group that could screen as possible anti-COVID-19 drugs. Thank you very much for your attention. And I would like to thank you once again for the opportunity you gave me to participate this work here.